Hello everyone, welcome to the third video of this metabolism and enzyme lecture. We all know about what are metabolisms, what is enzymes, what is give free energy, what is transition state, and we know that enzyme can help reducing the free energy of the transition state or activation energy, as you may recall. And the enzyme need to have a specific structure with the active site that can hold the transition state and convert it to make the products. So now I like you to focus on what are actually the proteins. Protein is just the final products from DNA. You start with DNA right here. DNA can replicate itself, we call DNA replication. DNA can be transcribed into RNA, or we can call it transcription. And RNA can travel from nucleus that is in nuclear envelope and get out, and then go to the ER where you have ribosome to make proteins, and that process is called translation. And these proteins can be a combination of many amino acids, and I will describe to you in just the next slide. So protein is the final product of the whole thing. Is called central dogma, and it is the most abundant macromolecules in all the cells, and it occurs in a great variety in both sizes and shape, and they have a lot of functions. So this is the basic unit of protein. It's called amino acid. It's actually composed of carbon, oxygen, hydrogen, and nitrogen. The main thing about amino acid is the alpha carbon in the middle, the carboxyl group composing of carbon forming two bonds with oxygen, and amino group or nitrogen with three molecules, three atoms of hydrogen that lose one electron. Another one is an R group or the side chain. This can be varied depending on the type of amino acid. There are totally 20 different types of amino acid, and their differences lie on the R group. This is an example of the peptide. Peptide is actually more than one amino acid connected to each other. And the bond that hold the two amino acids together is covalent bond. And this special type of covalent bond, because it's forming between two amino acids, we call it peptide bond. It's actually carbon and nitrogen from carboxyl group and from amino group. In the pink, you can see that they are not the same between different amino acids. Therefore, the pink color represents different R group or different side chain of amino acid. So you can imagine that if you have totally 10 amino acid in one peptide bond, and each position you can choose 20 different amino acid, and in just 10 amino acid length of the peptide, you can have a lot of varieties, because in one position you have 20 choices. All right, and normally in protein, in the peptide bond, in the peptide, each one of the protein, they have like 100 or several hundreds of amino acid. So you have a very endless variety of protein. And as I mentioned earlier, that different group composed of different chemical properties, different chemical bonds. So different properties, what I meant by that? I meant that some R group can be hydrophobic, some can be hydrophilic, some can have positive charge, some can have negative charge, some can dissolve in water, some cannot dissolve in water. So you have a lot of properties. That means that if you have a long length of protein, those chemical properties from different R group, they can form different interaction between the side chain. So they can adopt different structure. And in biochemistry, we divided structure of proteins into 4 forms. The first one is primary structure. It's just like amino acid connected to each other. It's more like a sequence of protein of amino acid connected to each other in only one dimension. All right. For secondary structure, you now have about 2 dimensions. The first one is the coil structure or alpha helix structure. So from primary structure, if you have interaction between the side chain of one peptide, you can form this alpha helix structure. Or if you have the folded of 2 strands like this, you can have the pleated sheet structure. It's more like a hydrogen bond between the side chain can interact with each other and form the 2 dimension of the secondary structure. The third one, the tertiary structure, now you have about 3-dimensional structure. It's the more like the whole length of peptide bond. They can form like a ball of noodles. And like this ball, far apart, the amino acid from far apart can interact with each other, forming the weak interaction. For quaternary structure, the fourth one, you can have more than one peptide bond, more than one strand of peptide. For example, you have the red one like this, and you have an orange one. They can form interaction with different balls. So we call each one of these as subunit. This is the first subunit. This is the second subunit. If you have more than this, we call it multiple subunit. And protein, when it function, it need to be either the tertiary structure or quaternary structure. That means it need to be in 3-dimensional structure. It cannot function if it is only 1 dimension or 2 dimension. They will not work like that. They need to be 3 dimension. But I fold this again. It can form like 3 dimension. All right. So this is an example. 3 dimension can hold the substrate, can hold the transition state of the substrate together. It can work that way. All right. And the protein that form a 3 dimension, we call that they have the conformation. Conformation is just an arrangement of atom in the protein that have different space. A protein in any of their function, it can have the native conformation. We call it a native proteins. That means if you not do anything with them, they will have that uh, shape without doing anything. They will form that shape that shape with by their native nature, by their nature. All right, the protein is stabilized. The different strand or different side chain can be stabilized by different type of weak interaction. For example, these two can. This is from one side chain and this is from another. They form hydrophobic interaction between non-polar side chain. They can form hydrogen bond between two polar side chain. They can form ionic bond, the negative ion and positive ion, between charged amino acid side chain. They can form disulfide bond between two cysteine residue. All right, and the protein is in native state. It can function very well, but this native state will be destroyed easily. It can be destroyed easily, and when it's not in native state, we call it is non-native or it is inactive. For example, the disulfide bond between the amino acid number 65 and 72, 26 and 84, 58 and 110, 40 and 95. These can be broken by many denaturation conditions. We will talk about this in the next slide. So the covalent bond between the amino acid is still there, but the weak interaction can be destroyed. And this is the unfold state. They will no longer form a ball anymore. They will be like primary or secondary structure again, and that cannot work. So what can denature the protein? Heat can break the interaction between, can break the hydrogen bond. Extreme pH can alter the net charge on the protein, causing repulsion, pushing, or disruption of hydrogen bond. Organic solvents such as alcohol or acetone can disrupt the hydrophobic interaction to make up the stable core of the globular protein. For example, you should not drink an alcoholic beverage when you have an empty stomach. Your stomach will be burned easily by drinking the alcohol. And finally, the solutes such as urea, guanidine hydrochloride, or detergent. This can disrupt hydrophobic interaction. Let's think about the heat again. If you cook an egg, if you notice the raw egg, you have the transparent egg white. But once you cook it with the heat, your egg white turn white now because it's no longer native anymore. You will find that the texture of the raw egg white and the cooked egg white is totally different. They lose their flexibility, they lose their fluidity, they will become more solid. This is the native form of albumin. They form coil structure, they form a globular structure. Once it's denatured, the weak interaction between the side chain is broken. That will that will separate the different strand of protein, and some cross-linking might happen in the case of the egg white. So you lose the properties of the protein, you lose the function. All right, we are now finishing the protein structure. So first you know that the protein is just enzyme is composing. Enzymes are protein, and protein to be function or enzyme to be function, they need to be in three-dimensional structure that they can be formed from the weak interaction between the side chain of amino acids, and the weak interaction can be destroyed. So that means your enzyme need to be in the native state. Your enzyme cannot be heated, cannot be in the extreme pH, cannot be in an organic solvent or some detergent. Otherwise, your enzyme will die. They will not be function anymore. So apart from the native structure of the protein, an enzyme, some of them need to have a helper. And those helper is just an additional chemical components called cofactor. The enzyme that just a protein is called apoenzymes. And when it bind to a cofactor, we call it holoenzymes. An example of this is the coenzyme A. This one. This is adenine, this is cysteine, this is the pantothenate, which are this coenzyme A is just a cofactor, or we call it coenzyme because it's a complex organic molecules. But the cofactor is the big set, and coenzyme is a subset of cofactor. Apart from coenzyme, you can have the inorganic ion such as irons, magnesium, manganese, and zinc. These ions can function as a cofactor. And example of coenzymes can be vitamin or organic nutrients. It's a transient carrier of specific functional group. And coenzyme or metal ion that bind tightly to an enzyme with the covalent bond, we call it prosthetic group. Example of some coenzymes or vitamin can be show up again in the photosynthesis and respiration lecture. We will stop here and we'll come back again with the next video about nomenclature and an example of it in the lab. All right, see you in the next video. Bye.